Good afternoon, everybody. Welcome to this June issue of My Story by AOS, supported by Plant and Food Research. We are very excited to welcome Lisa Matisoo-Smith from the University of Otago here today. I, of course, am not going to say very much because that's why we're here. We're here to hear Lisa tell her own story. So, Lisa, over to you.
Thank you, Emma, and it's very nice to be here. And I assume that there are people out there, even though I can't see you. Yeah, so I will just get going with my story. Ko ai au, o ko Surmunawagi te maunga o Okutupuna, ko te repo o Vasilina te wai o Okutupuna, no Estonia o Okutupuna. So just to explain, my family, on my father's side, is originally from Estonia, and Surunmunwagi is Egg Mountain, which is our family mountain, is the highest hill in Estonia, and the wetlands of Vasilina, which is where my father grew up, is the Y of our family, and actually they're the wetlands or swamp, and our name Matisoo actually means the swamp of Mati, so the swamp is very important to Estonians, and that's my background on my dad's side. My mum, Julia Merrill Moores Matisoo, is originally her family, her tupuna, are from England, Scotland and Wales, and were some of the first people to head off to the New World and the Americas in the 1600s. And they landed on the East Coast and eventually moved to Indiana, which is where my mother met my father. My father was a refugee after World War II from Estonia, and his family ended up in Indiana. So that's where Mom and Dad met. We're high school sweethearts. And yeah, Dad joined the U.S. Navy after studying engineering at Purdue University. And as a Navy family, we didn't travel as much as most. I was actually born in Hawaii. Dad was based at Pearl Harbor. And I lived in Hawaii until I was 8, at which point we moved to Japan. And I grew up and went to school on a military base, but we traveled as much as we could and lived off the base at various points of time in Yokohama, Japan. So I finished my schooling there in a Department of Defense school and decided to head off to university. And after a brief sojourn at Boston College on the East Coast, I ended up realizing that I really wanted to be at the University of California at Berkeley. And that is where I did my undergraduate degree in anthropology. So I am an anthropologist by training, and as an undergrad in the US system, I was trained in 4-field anthropology. So I studied archaeology, linguistics, cultural anthropology, and what we called back then physical anthropology. Now we call biological anthropology, which was my specialty, or the area that I was most interested in. I loved this field because it was really a mix of the social sciences, and I really loved the biological sciences. Those were my main areas of interest. And so we actually had data, which was always kind of my frustration. My mother was a social anthropologist or cultural anthropologist by training, studying while I was going to school in Japan. She was getting her master's degree in Japanese psychological anthropology, and I liked the data of biological anthropology. It was more sciencey. And so I decided that I wanted to be a biological anthropologist, which is not a career that many people think of or know about, even as a subject. But I was fascinated, and many people have suggested, perhaps it was my upbringing, living in different cultures, that drew me to anthropology. But basically, biological anthropology looks at humans, and we really are trying to understand and explain human variations. That includes everything from origins and hominin origins, human evolution, where do we come from, how do we get here, the environment, understanding the relationship between humans and their environmental stresses and how we adapt to those in a number of different ways. Looking at diet and how, again, that contributes to who we are and our variation. understanding the cultural traits. Who do we marry? What kind of clothes do we wear? What kind of housing do we build? What kind of activities do we undertake? What is our role or status in the group? All of these things affect human variation. And of course, interactions and migration. So as an undergraduate, I was very lucky at Berkeley to be studying with Tim White and Don Johansson. Many of you will have heard of Lucy, Australopithecus afarensis. I really wanted to go to Africa and study hominin evolution, but I was pretty much told at the time as a woman and as a white woman, it was unlikely that I was going to get graduate research in Africa. They were really at that point focusing on training African researchers, and that perhaps I would should consider looking elsewhere to study human evolution. So I decided I would head off to Europe and study Neanderthal, and wrote to get kind of fieldwork opportunities around Europe, wrote off to various projects that were taking place, and was accepted to work at a very famous Neanderthal site called Lazare, that is very close to another very famous site called Terra Amata, which is the first evidence that we have of human modification of a cave. Those are both in Nice in France. And so after I graduated from Berkeley, I took off to Europe with a girlfriend to backpack for a few months, and then to go to work at the site of Lazare with the Institute of Human Paleontology. But on my way overseas, the first stop we made, the first day, we landed in London and stayed at a local youth hostel and asked where the best pub was, and we were directed down to a pub called the Baynard Castle, just on the Thames there, and walked into that bar and ordered a Guinness, a pint of Guinness, and met lovely Kiwi barman who served me that pint of, well, actually served me 2 half pints of Guinness, because the story they told me they didn't serve women pints of Guinness in the pub. So I said, then give me 2 half pints, and he stepped up, and we ended up talking for the rest of the night and the next few nights that we were in London. And yeah, I actually ended up marrying that barman, not just in those first few days, but he wrote to me while I was traveling with my girlfriend, and eventually came down and met me in Nice, and we are still drinking Guinness to this day. So my lovely Kiwi husband, Brent Smith, there is basically why I ended up here in Aotearoa. So we got married. We met in '86. We got married in '86, and I started. Actually, we met in '86. Got married in '87. Sorry, and I started at the University of Auckland soon after that. I decided I wasn't going to study Neanderthal. I came to Auckland and met people at the university to talk about what I might be able to do. And it really kind of brought me back to my Pacific roots when I realized there were a number of questions that were still unknown about the settlement of the Pacific. And I had the opportunity to start thinking about the latest kind of tools that we used in biological anthropology, which was using molecular tools, or trying to understand human evolution from a genetic perspective, which is actually a fascinating field that was developed by a New Zealander, Alan Wilson, who was at the University of California while I was studying there. Unfortunately, he passed away in 1991. But yeah, so I was really interested in the Pacific. But while I finished my master's thesis on genetic variation in Polynesia, I was also working on another major project, and she was due the same day as I handed in my master's thesis. Thankfully, she was 2 weeks late. Our daughter Tessa was born in November of 1990. So I juggled her and a part-time job teaching in the department, and then decided to enroll for a PhD, applying the latest molecular tools to understanding the settlement of the Pacific. So I was very lucky. I had 2 wonderful supervisors, Professor Roger Green, who is a Pacific prehistorian, was also very interested in a multidisciplinary four-field approach to understanding human history. And Professor David Lambert, who took me under his wing and into his lab and set me up to learn about evolution and genetics and genomics and the tools that I have now applied for the last 30 years to my research. So two very wonderful mentors. Dave Lambert is still is now at Griffiths University in Australia, and unfortunately Roger Green passed away. But sitting next to Roger there is another colleague of mine who was my kind of academic big brother, Richard Walter, who is a professor of archaeology here at Otago. And so my PhD research really was focusing on reconstructing the human settlement of the Pacific and using molecular tools to try to do that. And as I say, Alan Wilson's work in the late 1980s and early 1990s focused on looking at mitochondrial DNA variation, and I was wondering about applying that method to trying to understand the Pacific. But in the 1990s, that was, for very good reason, a very controversial topic, using human DNA, particularly from indigenous communities. And I was very lucky in one of the people I was actually teaching with. I was teaching human osteology at the med school, and Dr. Mere Roberts was teaching at the medical school, running their medical program, their first early ELM program, and she was doing her PhD on at the time on the gut parasites of the Kiore, or Rattus excellens. So she was telling me about the Kiore and how the Kiore, which is not native to Aotearoa, how it got here. And in those conversations, we started thinking about the value of looking at this animal as a tool for actually tracing human migration. So Mere and colleagues explained how the Kiore were transported intentionally by people in the waka that came down here to Aotearoa. And in investigating this and the archaeological record, I could see that these rats were transported from their homeland in island Southeast Asia with people and appeared in the earliest archaeological record throughout what we call remote Oceania. So people carried plants and animals that were important to them in their canoes and introduced them to the new islands that they were settling across the Pacific. So I developed what became the topic of my PhD dissertation, what we call the commensal model, by using the DNA and understanding the DNA relationships of the plants and animals across the Pacific, and using them as a proxy to track the movement of the humans. So the rats only got here with people if we could figure out where the rats came from. Those that were brought to Aotearoa, that would tell us where the waka came from, and therefore where the people came from. So along with the rats, people also carried pigs and dogs and chickens and a number of plants. And we've now, over the years, extended the commensal approach to looking at a number of species intentionally and unintentionally introduced by or with people. And as a result of those conversations and understanding that Kiore were intentionally transported to Aotearoa, let's see if I can move that. I was introduced to another incredibly important person in my career, and has become a mentor and dear friend, Mr. Hore Parata from Ngāti Wai. And this is to say the early 1990s was when the treaty claim, Y262 claim, was filed by 6 iwi, including Ngāti Wai, for control over indigenous knowledge and plants and animals. So I had a very rapid and steep learning curve about the importance of Kiore, and about working with communities and consultation. and I have learned so much that's been so valuable and has contributed so hugely to my research, both the science and personally thinking about how I undertake research. So really valuable getting to know Hori. Now what's happening here? There we go. And Hori and others explained to me that Kiore were intentionally transported. They are taonga, certainly for Ngāti and other iwi as well. And this is a beautiful carving from the whare at the University of Auckland, Tane Nui Arangi, of Ruanui, the navigator with the Kiore traveling on his shoulder. So a very different perspective to a Western, a typical Western concept of rats are vermin, rats are bad, people wouldn't intentionally take them. We have a very different, recognize that there's a very different relationship and different way of thinking about these animals and many animals, and that's something we have to consider in any commensal model. So this was a wonderful kind of reenactment of the transport of kiore in little waka kiore, little containers that they could be transported in. We've had many debates over the years as to whether or not there were stowaways, but there certainly is information about the intentional transport of kiore and behavioral characteristics that indicate that they probably were not unintentionally transported, and certainly our data have supported that. It was explained that they were important for a number of reasons, but including that they were an important food item, important for gifting in parts of the motu. They were often preserved in fat and gourds and were gifted to chiefs or to particular high-ranking people. And I've looked at this information from across the Pacific, and it varies across the Pacific, but certainly they were an important food item, and we do see that in the archaeological record as well. And so in talking and establishing these relationships and starting this consultation of working with iwi, with communities on species that are of significance, it brought up a lot of issues. And this was very early on, but these are issues that we still deal with today, and doing any genomic work and any archaeological work as well, with communities generally, but certainly with Indigenous communities, about how do we actually start consultation. And we had set up a consultative committee that Hori directed. But this is something, how do you, different iwi are interested in different species, different rohe overlap, and these are issues that we're still working through today. Similarly, the ownership of knowledge and recognition of that ownership of knowledge and the ownership of data that are generated from taonga species, for example. And we did this very early on. I contacted GenBank. Hori had asked me who would have access. We decided to look at mitochondrial DNA of the kiore to track their migrations. And as part of the publication of any genomic data, we actually have to submit that data to a database, a public database. And Hori wanted to know who would have access to that data and what they might be able to do with it. So we wrote to GenBank and asked if we could limit who had access to that data. This was in the mid-90s by this time. GenBank came back and said, no, it has to be public. So we had consultation about that and answered questions about what people might be able to do with that mitochondrial genome data that we submitted. We talked about the interpretation, what we thought the data meant that we found, and what iwi might interpret from those results, and how we might communicate that information, and how we might really develop a partnership that was of mutual benefit, in particular of any research that was of relevance to Maori. So these were really important lessons that we started working on very early on, and there are no simple answers, and these are still questions that we're dealing with today. But in 1997, I finished my PhD. We published a paper the main paper from my thesis in PNAS, which was a real thrill for me and for the team, that showed how the Kiore had been distributed around the Pacific in the Waka. So we had settlement from West Polynesia, Samoa, and Tonga to Central East Polynesia, the Society Islands, it's Tahiti, Raiatea, Bora Bora, and the Cook Islands. And from there, we had two major interaction spheres, a northern sphere where rats were taken through the Marquesas and up to Hawaii, and also directly up to Hawaii, out to Rapa Nui, Easter Island, and down here to Aotearoa, with the Kermadec Islands appearing as a bit of a stepping stone, and ultimately out to the Chatham Islands. So this was the topic of the first major paper of my PhD, but it also kind of put me on my career path, I suppose. I was able to get some funding for a postdoc through Forced, as it was at the time, and Marsden grants. And we expanded this commensal approach to address not only extant or fresh samples of Kiore that I trapped from around Polynesia, but moved back into the Western Pacific to really look at the origins of Polynesian rats. And we were able to also then apply the latest techniques that allowed us to extract DNA from ancient remains or from skeletal remains, from archaeological samples or from museum samples of rats. And so I extended the project into the Western Pacific. So we had just kind of one lineage of rats out here in Polynesia. But when we studied the rest of the moved back into the Western Pacific, we found out that there were a number of different types of rats. Some closely related rats, these we call the group 3 rats out here in Polynesia. We found some 3A rats here in the kind of mustardy color. But then we found two very distinct different groups in what we call near Oceania and in island Southeast Asia. So we didn't see any really direct links between Polynesia and what we expected from the archaeological and linguistic and cultural data back to near Oceania, which is thought to be the place where the Lapita people, the ancestors of Polynesians, first appeared. But in looking, so this is a map of the Lapita dispersal about 3,350 years ago. The Lapita culture appears in the Bismarck Archipelago, and within a few hundred years, they moved out into what we call remote Oceania, where they were the first people to settle these islands. People have been living in near Oceania for tens of thousands of years, but the islands of remote Oceania were first settled by Lapita people about 3,000 years ago, or 3,100 BP here refers to years before present. It's an archaeological way of dating, or 3,100 years ago. We see evidence of Lapita sites in the Reef Santa Cruz and Southeast Solomons, in Vanuatu, New Caledonia. Around 3,000 and 2,900 years ago, we see evidence of people in Fiji, Tonga, and Samoa on the edge of the Polynesian Triangle. But Lapita expansion stops there, and people didn't move into East Polynesia for another 2,000 years or so. But what I realized when I was looking at the data that we had generated for near Oceania was that none of the samples that I actually had in my study came from Lapita archaeological sites. And so I decided, and I wrote a Marsden grant and decided we really need to go out and sample rats from those sites in the Bismarck Archipelago that had archaeological evidence of early Lapita sites. And so I was very lucky to work with a colleague, Glenn Summerhays, who I've worked with for many years. I was an archaeologist now at University of Otago, and our colleague, Mr. Herman Mandui from the National Museum and Art Gallery in Port Moresby, to go out and collect rats from islands that had evidence of Lapita occupation in the Bismarck Archipelago. So we had a very targeted sampling strategy, and I learned, again, hugely valuable information about consultation from Glenn and from Herman. And we kind of had some challenges that were quite different to working to the sampling that I was doing in Polynesia. Because there were a number of sites that women are not generally allowed to visit for safety issues. Women are associated with magic and so forth, and are limited in places that we can go and when we can go to those places. So we actually, through consultation with the communities, realized that we had to get the community involved in the actual sample collection, so the trapping of the rats, because the local people knew where it was safe for people to be and where to be trapping. So I would hand out rat traps to people and explain to them in little vials of ethanol how I wanted them to take a bit of a tail snip and an ear snip and a foot and put them in the ethanol, and they would either bring those samples back to me or bring me the dead rats. So there was much hilarity in that process. People thought it was very odd that I was interested in rats and what did I really want to do with them. So lots of discussions about why I was studying them. It also gave an amazing opportunity for education and outreach. So people would bring me the rats. I would go off to the side of the village and start recording what species they were, because there are many different species in the Bismarck Archipelago. And I was only interested in Rattus excellens, the Chiore, which you actually can most readily identify by the number of nipples that they have. So again, people thought that was pretty funny that I was counting nipples. The Chiore only have 8 nipples and the other rats have 10 to 12. And then I would take measurements and other characteristics. So the kids would all start coming around and watching what I was doing, and I'd be talking about what I was doing. And pretty soon they were identifying what species were there and whether they were male or female. And they started wanting to take the samples, and it became quite a community event. The kids would get involved, and then the women would come around, and then the men would come around, and pretty soon it became a bit of a competition who could get the most rats and who could get the biggest rat and who could it certainly was a lot of fun. But then of course people actually wanted to know what the results were. So when I would return in previous years, everybody was very excited to hear who got what kind of rat and so forth. We also did with Glenn a lot of school talks. He has a very long history of working in New Guinea and wonderful relationships. And we would do radio interviews and talk about the research that we were doing. He was talking about archaeological research and I was talking about the DNA work in the rats. And during one of these interviews, a couple of school teachers heard our interview and heard Glenn talking about Lapita archaeology and Lapita sites, describing the most identifiable feature, which is this beautiful dentate stamped pottery that is the most notable marker of a Lapita archaeological site. And they said that they thought that they had a Lapita site on their island. On the island of Emera, which is a very small island, about as far away as you can get from Port Moresby, which is down here. And so they invited us to their island in 2007 to investigate this Lapita site that they had identified. They sent us photos of the pottery, and indeed it did look like it was Lapita pottery. So they had excavated the site, which made us a little bit nervous, but we did arrive on the island in 2007 when we're taken out to the site. They decorated the site ready for us. I always tease Glenn about how hard it is to find a Lapita site when it's presented to you like this. But we put down some archaeological pits next to where they had excavated, and they had recorded at what depths they started finding different things, the shells and pottery and so forth. And they had indeed found a very important Lapita site, turns out to be the earliest dated Lapita site now. And their archaeology was surprisingly good. And their recording of all of the information was outstanding. So we extended the excavation over a number of years. The kids got involved, the community got involved in the excavation, which included also my trapping rats and looking for rat bones in the archaeological site. So wonderful community involvement. And over a number of years, we went back and re-excavated, returned results about the rats. And the community were very interested in these results, but they said, we get that you're looking at our origins through the rats, but why don't you just look at our DNA? Wouldn't that tell you where we came from and how we're related to other populations? And so there had been quite some change over those years in building those relationships where there was trust, and the community was actually interested in understanding about their own DNA. And so it was about that time that I was approached by National Geographic, who were starting what they called the Genographic project, and trying to understand human migrations worldwide by looking at human DNA, mitochondrial and Y chromosome DNA. And while up until that conversation with the community in Emerau, I would not have thought that Pacific communities were keen on engaging in this research. But with the request from Emerau, I said to National Geographic that people might be willing to engage from the Pacific, and I would be willing to do that if those communities, if it was community-driven research. So National Geographic took me on, and I became one of the principal investigators. I had to point out to them, first of all, that their maps were really not great from a Pacific perspective, that the Pacific Ocean is not just a blank space where you can put your key of your mitochondrial Y chromosome lineages, that was covering over many of the islands that I would be, and the communities I would be working with, and they really needed to have a more respectful map in their study if we were going to engage the Pacific. So it was also at that same time, 2008, 2009, that I started having conversations with the University of Otago. I had, after my postdoc, been offered a position in the anthropology department at the University of Auckland. So I taught there for many years. But in 2009, I was made an offer I could not refuse by the University of Otago, and came down and was in fact working with Glenn Summerhays and Richard Walter, two of my close archaeology colleagues. But I joined the Department of Anatomy, which is where biological anthropology is, or was, at the University of Otago, and still is today. And that was a real shift. It's a wonderfully multidisciplinary department that includes lots of biological anthropologists, but also neuroscientists, clinical anatomists, developmental, reproductive development, and genomics researchers. So it was a wonderful place to come and work. So I continued my fieldwork in Emerau, went back, and we, as part of National Geographic, started collecting cheek swab samples from the community. And started talking to them about mitochondrial DNA and Y chromosome variation, and who we needed to sample in order to get all of the variation, maternal and paternal variation. Mitochondrial DNA follows your maternal line, your mother's mother's mother. You just get your mitochondrial DNA from your mother. For men, they get their Y chromosome data, their DNA from their father, and so Y chromosome will trace the paternal line. So we explained this to the community. We had been looking just at mitochondrial DNA for the rats. It's the same system there. So they amazingly pulled out their genealogies, which they had recorded. The island, the entire island is Seventh-day Adventist. They all belong to the same church, and the church is very strong in recording of genealogy. So they had all of the information there, and they could identify very quickly how I could sample the entire island and make sure that I had all of the maternal and paternal lineages on the island. They were also very interested in answering some questions that they had. They wanted to know whether there was any correlation between mitochondrial DNA and their clan system, their clan structure. So we modified our sampling information forms to include that information as well, so that we could answer that question for them. And as it turns out, yes, it did. So we sampled the island almost completely, about 450 people on the island, and we sampled what we believe was all of both mitochondrial and Y chromosome diversity on the island. And in 2009, it just so happened that a nearby island, about 5 hours by boat, a low-lying atoll called Tench, had been flooded in a king tide, and the whole island was evacuated to Emerau while the island recovered. And so we were also able to sample the Tench community there. And the Tench community had not actually recorded their genealogies, but when we explained how things were working with the Emerau community, they realized that they really needed to start recording that information before it was lost. And so this gentleman here was the last person who knew most of that genealogical information. And so they sat down and started recording as best they could the genealogical history for that island. So that was a really lovely opportunity, and we were able to sample that community as well. So we, as part of the Genographic, we sampled not only Emerau and Tench, but a number of other Pacific Island communities and locations, Tokelau, and throughout the Society Islands, working with colleagues in French Polynesia. And we did both modern DNA sampling in Tokelau, but also ancient DNA sampling in Tokelau, looking at mitochondrial DNA. I was working with an archaeologist, David Addison, who had we had a major NSF grant there doing excavations and biological anthropology, recovery of skeletal remains with my colleague, Frédéric Valentin from Paris. And so we were excavating as well as trapping rats, and as well as sampling human taking swab samples for National Geographic. And the community, one of the communities, the community of Atafu, were really interested in the ancient DNA, and they wanted to know whether or not they were directly related to the people who we were recovering from the archaeological excavations, because their oral traditions talked about other people being there when they arrived. And so they requested ancient DNA analysis. One of the other islands, Nukunonu, did not want ancient DNA analysis done, so any human remains that were recovered were reburied on the island. So we were able to look at the mitochondrial genomes, and interestingly, we found that everything from about 45 or 35 centimeters and above all belong to one mitochondrial lineage, which was the lineage that we actually saw in the people of the island today. But the deeper samples had a slightly different mitochondrial lineage. And so their oral traditions, sorry, got those the wrong way around. These were the higher levels before A1A1, and those are the lineages that we see in Tokelau today on Atafu. And these B4B lineages we did not find in the Atafu population today, but they are common in Tuvalu and in places in Micronesia. So their oral traditions turned out to be right, and that was pretty interesting, that it looked like we had replacement on the island of Atafu, probably from Samoa, which fits well with their oral traditions. So in having those close relationships, both collecting the genealogical information and then being able to go back to the community the next year with the data to talk about the results and give the results back to the individuals and to the community, it allowed us to solve several mysteries, I suppose. We found, for example, an individual who carried an L3 mitochondrial DNA lineage in Tokelau, and the L3 lineage is an African lineage that really we tend to find in African populations, or recently derived African populations. So we certainly did not expect to see it in the Pacific in Tokelau. If it was a Y chromosome, we know sailors were moving around historically. That wouldn't be unheard of, but certainly from mitochondrial lineage, it was a surprise. But in going back and talking to the families, we solved that. It turned out that the mother of the person who provided the sample was adopted from Hawaii, and she had been told that her mother was of Puerto Rican ancestry, which is not uncommon in Hawaii, and African ancestry in Puerto Ricans is also very, very common. So she was delighted to understand her heritage, and that solved our problem, our question as well as to how did this L lineage get into Tokelau. So you've really got to engage with that history, recent and deep history. So this is return of results to people, and this, we gave posters and things to the schools. Similarly, we found something in Emara over the years of sampling, that we found some lineages that were quite rare, that were found at only 4% on Emara, and we were able to trace those back to a particular woman who came from Manus Island. So that was not that was not an ancient lineage on the island, but was a more recent ancestor, but beyond the 2 generations of information that we were collecting. So really important to have those relationships. And it was at about the same time that I also started working at this amazing site here in Aotearoa. I'm working with the community around the site of Warabar, which is your classic moa hunter site located at the mouth of the Wara River, just outside of Blenheim. And one of the most important sites in New Zealand, where we have one of the largest burial populations. So in 2009, just as I came to Otago, we started a conversation and negotiations with the Canterbury Museum and Rangatane o Wairau, the iwi, to discuss the return of material that had been collected through archaeological excavations, and some have described as grave robbing from the 1920s onwards. And that involved discussions about the possibility of biological analyses of the tupuna that were in the Canterbury Museum prior to their reburial back on the site of Warabar. So Warabar is one of the best dated sites we have in New Zealand. Very approximately dated to 1320 AD, and for a duration on the site where excavations were taking place of 25 years. So very precise. And Rangatane, after many conversations and discussions and presentations, decided that they would agree to and support biological analyses of the tupuna before they were reburied. So samples were taken. They actually asked us to take some samples for archiving, given that DNA techniques and technology was changing quite quickly. And then the tupuna were reburied on the site. So we've been working for many years now with Rangatane on the ancient DNA of those tupuna. In 2012, we published the complete mitochondrial genomes of several of those individuals, and we've continued working on those. We now have, I believe, 12 individuals sequenced for complete mitochondrial genomes, and also now whole genome, so nuclear DNA analyses as well, so we can start to address even a number of other questions, not just the origins of populations, but the size of the population, the size of settlement population, and even looking at some of the health issues. We were thrilled in 2016 that Rangatane hosted the New Zealand Archaeological Association annual conference, and welcomed everybody onto the site to talk about the site and the history of the site. And the community, because we had presented the results of the ancient DNA, the Rangatane asked whether they could participate in the Genographic project, and so that we could compare their DNA to that of the tupuna from the site. So we started looking at sampling members of Rangatane today. So all of this data combined, the rats, the people, the archaeology, the linguistics, the cultural anthropology, ties in to help us understand the settlement of Aotearoa. And as I say, we've been working, looking at whole genome data now of both the ancient and the modern samples, really trying to understand the complexity of the settlement of the Pacific. And it is turning out to be more complex than we actually thought, particularly the relationship of the Lapita people with the people in Near Oceania and as ancestors of the Polynesians. And in looking at this whole genome data, we also realized that we could start to address some of the health issues that are affecting Pacific peoples at higher frequency than others. And I started working with a colleague, Tony Merriman, in the biochemistry department here at the University of Otago, who was looking at the evidence of gout and metabolic disease in the Pacific, where we have much higher rates of gout and metabolic disease. And they've identified a number of genomic markers associated with the likelihood of being impacted by gout and metabolic disease. And so we are now using the archaeological, the biological, anthropological information to try to look at the origins of metabolic disease in the Pacific and trying to understand why Pacific peoples have such high rates. It was always associated with adoption of Western lifestyle and diet and lack of exercise. But we've been able to show that we actually have evidence of gout in the earliest archaeological records, including at Wairabar, but in the Cooks and Hawaii and in Guam. So this is not a modern disease. This is something that Pacific people have been suffering from for thousands of years. And so we're trying to understand the origins of that and the evolution, what's causing the high rates of metabolic disease in the Pacific, and hopefully change the discussions away from a very, what has been a Western kind of finger pointing, you're eating the wrong foods and not exercising enough, and that's why you have gout, diabetes, obesity, cardiovascular disease. That recognizing that this is actually a genetic predisposition and it may be related to selection in the past. So we're working with colleagues at the University of Papua New Guinea and the University of Guam, and with colleagues here at the University of Otago. So it looks like I'm getting close to running out of time, but very quickly, what we also carried on and looking at kind of using DNA to understand human migrations, to not just apply it to the indigenous settlement of the Pacific, but we realized this was a brilliant way to engage communities in understanding science, DNA, history, and so trying to understand the history, the full history of humans and settling in the Pacific. I was able to get National Geographic to allow me to extend my study to all New Zealanders. And I started a project called Africa Te Aotearoa as part of a Cook Fellowship from the Royal Society and funded from National Geographic that would involve citizen science and engage the public in tracking our national identity. Where did we all come from? Because we all started in Africa, and Aotearoa being the last place that was settled by humans, the story of New Zealanders really does tell the human story as a whole. So we conducted random sampling across the country in all the major cities, 5 major cities, and some of the smaller areas as well, just randomly sampling people on the street and collecting their cheek swab DNA, and then returning those results to them. We did marae-based sampling as well, and community sampling, for example, the Otago Chinese community and the Dunedin Lebanese community, the Auckland Dalmatian community, to look at their DNA as well. And we've got some amazing results. We were quite surprised, the similarities between Christchurch, Wellington, and Dunedin, almost identical mitochondrial signatures there. But those were quite different from Hamilton and Auckland, for example, where we see much higher levels of mitochondrial haplogroup B, which are Polynesian lineages. These other HT, U, K, and J are European lineages. M's are generally South Asian lineages. Those are the L are the African lineages there. Fascinating results when we broke down Auckland into different neighborhoods, for example, quite a lot of variation, which was and wasn't surprising. Parnell looks the most kind of standard Western European, and we have much higher rates, for example, of the Pacific lineages in Otara, but also in Onehunga. Interestingly, the L African lineages, high frequency in Glenfield, which we know we have large South African populations there living in Glenfield. So it's fascinating to look at where people settle and all of those issues. But basically, we found all of the major mitochondrial lineages that exist are found in New Zealand, in New Zealanders. So we really do tell the whole story of human history, really. So lots of lessons learned about consultation and the value of that, hopefully for all involved, the needs for conversation, for trust, for engagement and real partnerships, and the value and importance of multidisciplinary research between sciences and the humanities. So I think this is my final main slide. People, I'm an anthropologist. It is the most important thing. He tangata, he tangata, he tangata. And just very quickly acknowledge the funding and institutional support. And I've been very lucky. Both my husband, Brent, and my daughter, Tess, have joined me in the field and helped me as field assistants in sampling, and wonderful colleagues that I work with. And of course, a huge team of postdocs and students and everything who've contributed to this as well. So I will leave it there. And it looks like we have some questions.
Yeah, thank you very much. I was intrigued that Anne Geddes took a picture of you with your baby, so I'm hoping that there's a picture of her in a flower pot somewhere.
There is. She was the first flower pot baby. Anne knew that she was about the right age. She was 3 months old when we had that photo taken. She wanted babies who could sit up but not yet pull themselves up for the big flower pot series that she did. So Tessa was one of those babies, and they ended up not being able to do the thing. So Tessa is in the lineup of flower pot babies. She's yeah.
Yep. Yep. I think anyone who was a teenager in the 90s had a flower pot baby at some point. So,
yep.
We do have a couple of questions. So first one would be, what is your opinion on ancestry websites and services?
Yeah, you have to be careful, be very careful, because that data can be used for all kinds of things. Companies are sold, there are breaches of data. So what data are you making available? What's publicly available? What's not publicly available? There are issues about, if you're just looking at mitochondrial DNA or Y chromosome DNA, that's not problematic, although the databases that they use are problematic. So the information that they give you of being a particular percentage of this ancestry and a particular percentage of that ancestry is very variable. So you will get different results if you send it to different companies, or even if you send your sample over time, you'll get different results. Obviously, your DNA isn't changing. It's the database that they're comparing that DNA to. So you have to be careful there. If you're going for the whole genome stuff, again, I would be very careful and hesitant, I think, because we still don't have very good legislation over the use of that information. So we're trying to get that under control. But if you know that you have predisposed to particular health issues, that could have issues with insurance, with getting health insurance. So, are you doing it for a bit of fun? That's okay, but I wouldn't take it too seriously. And I would be very careful about what information I was happy to have out there publicly available. So,
yeah. So another one, you mentioned a few archaeologists who you looked up to and have worked with. Do you have other mentors or people who have inspired you to progress in your career?
I've had many mentors. Again, my mentors, both academically, but also in terms of my career, Roger Green and Dave Lambert, Mary Roberts, Hori Parata, all of those people. But yeah, let me think of others. I mean, other women who have really paved the way for us, I think. When I look at those people, Rebecca Kahn, who started the mitochondrial DNA work, but also, many senior academics, Carolyn Burns, for example, emeritus professor, did such amazing things as a woman and for women. Yeah.
I actually had a question. You mentioned a couple of times around not not being able to get funding to go somewhere, or being told you couldn't go somewhere because it was dangerous because you were a woman. Is that something that you think has changed? Was it like a genuine danger, or was it just a perceived danger because there were so few women in the field? Or has it shifted a lot in your career about where women can go?
Yeah, I mean, interestingly, in my field, in kind of studies of human evolution, some of the groundbreaking people have been women. And, if you think of Jane Goodall and Diane Fossey and Birgit Galdikas, they were chosen, Richard Louis Leakey chose them because they were women, and he thought women were better at observing than men, kind of classic things. But in the field of paleontology, other than Mary Leakey, there had, women were much fewer in number. I think there was initial concern, certainly in going to Africa, and the concerns of taking women into really isolated locations. I mean, I found more kind of not so much issues, but people raising things like, how could I go into the field with a young child? Who would look after my child if I was going into the field? And it's like, she has a father. I tried to get funding, a bank loan for my first as a graduate student, trying to buy a computer, and was told that anthropologists didn't need computers. And what did I have, what would I need a computer for? And what would I have as collateral? I said, my car. And the bank manager said, well, that would belong to your husband, not you. And it was like, no. So, I mean, and that was in the 80s. Yes, I think things have changed. I think we do have to think about our safety in the field, which sadly, I think we have to think about that more seriously than many men do. I don't think it's an issue with funding, or certainly I haven't experienced it as an issue with funding. But you do have to think about keeping yourself, keeping students safe. And in many places in the world, even here, it's there are issues that we have to think about as women that men don't have to think about.
Yep.
And it's something my colleagues, particularly the archaeologists, female archaeologists, we do talk a lot about that, and how we can talk with each other and share information to make sure that we and our students are safe.
That probably brings us to the end, I'm afraid. So thank you very much, Lisa, for talking to us today. It's really interesting stuff. The idea of bringing rats intentionally, that's the thing I'm taking home today. Thank you to everyone attended. Thank you to Plant and Food Research for their support of this series. Next month, our My Story will be Sila Wehi, who's the co-director of Te Pūnaha Matatini. That's going to be on the 4th of July, same time, same channel, as it were. And I just wanted to let everybody on the call know that Friday we will be celebrating Dell Day. So this is a celebration of women in science, marked by what would have been the 100th birthday of Dame Miriam Dell, who was the AWIS patron. So there will be an online celebration between 10:30 and 12. We're hoping people will get together and maybe have a bit of morning tea or a bit of lunch before or after. Get in a room, watch the online celebration, some talks by different women in science in New Zealand, talking about their science heroine. Flood your social media with your science heroine stories. Tag everything #science heroine or #DellDayNZ. And we hope to see you there. Thank you all for coming. Keep an eye on the website for the details of Sila's My Story, and we will see you next month. Thank you.
Thank you.